Today's assignment is on cells and the cell theory. The objectives for the lesson are to list the scientists who contributed to the cell theory, list the components of the cell theory, compare prokaryotic and eukaryotic cells, label a plant and an animal cell, and know the functions of the cell organelles. I know that all of these things are going to be tested, so make sure you pay attention. You might want to rewind and listen to parts of this presentation more than once. Robert Hooke and Anton von Leeuwenhoek were two of the first individuals to see cells, fairly close to each other in as far as time goes. Robert Hooke was the first person to officially see cells. He was looking at cork, which is a plant that makes up, we use it for bulletin boards and sometimes sandals and things like that. And he noticed that there were a great many boxes, as he called them. And you can see his image that he would have seen under a microscope. And so, since he saw all these boxes, he called them cells, like cells in a prison. Leeuwenhoek observed living cells in pond water, so he actually took water with his primitive microscope, and he could see things swimming around in there, and he called them animalcules. And that was 8 years later after Robert Hooke. Then we have Theodor Schwann and Matthias Schleiden. Schwann was a zoologist, so he observed tissues of animals and noticed they had cells. Schleiden was the botanist, and he noticed that plants also had cells. And then Rudolf Virchow reported that every living thing is made up of vital units, which he called cells. And he predicted that cells come from other cells. Notice this is more than 100 years after Robert Hooke and Leeuwenhoek. So the cell theory was kind of come about through a variety of scientists. The first part of the cell theory is that every living organism is made of one or more cells. Everything that's alive must be made of cells. Remember, we learned that way back at the beginning of the school year when we were talking about the characteristics of living things. Second part is that the cell is the basic unit of structure and function. So this is how every living thing is organized. It's organized according to the cells that are in it. Next is that the cell is the smallest unit that can perform life functions. Those life functions, remember, characteristics of life, able to reproduce, able to grow, develop, etc. And the last part of the cell theory says that all cells arise from pre-existing cells. So a cell doesn't just appear out of nowhere. It has to come from an existing living cell. Things that all cells share, features or organelles that all cells have, are ribosomes. Ribosomes are the protein factories that make the protein used by the organism. Remember that every living thing basically runs on the proteins that their body makes, or that the organism makes. All cells have cytoplasm, which is this fluid material within the cell. It acts as a protective area. It also makes sure that the environment is correct and in the correct pH balance and things for the chemical reactions that have to happen inside the cell. All cells have some sort of DNA. It might not be found in the same place. It might not look exactly the same, but this is the genetic material that gets passed from the parent organism to the offspring. And all cells have a cytoskeleton, which is sort of the internal framework of the cell. It keeps the cell's shape. And last but not least, all cells have a cell membrane. This is some sort of outer boundary where it allows some things to go into the cell and some things to get out, but not everything. So on your assignment, you'll see these 6 true or false questions. Number 1, true or false, was Robert Hooke the first person to see cells? Number 2, bacterial cells have a cell membrane. Think about what we just talked about. Number 3, the cell theory was developed by a single scientist. 4, plant cells have cytoplasm. 5, cells taken from fungi do not have DNA. And 6, cells can only come from pre-existing cells. Once you've answered true and false to each of those, then move on. Let's talk about prokaryotic cells, or prokaryotes. This is an example, a diagram of a prokaryote. Take a look at its features. Notice it has ribosomes, those are the things that make protein. It has DNA, it has a cell wall, and it has a plasma membrane, which is the same as the cell membrane. It also has a flagellum. Prokaryotes were thought to be, or are thought to be, the first cells to inhabit the earth. These are very simple cells. They don't contain a lot of complex structures. Bacteria is an example of a prokaryote, and their main distinguishing feature is that these cells do not have a nucleus. Their DNA is circular and it floats around in the cytoplasm. It does not have a package to contain that DNA. Some bacteria, but not all, have this flagella, which is a tail-like structure that helps it move. And some bacteria also are surrounded by a capsule, which basically allows them to survive through the body's immune system to some extent. It's a protective thing. Here are some examples of bacteria, which again are prokaryotic cells. They do not have a nucleus. You'll notice here in this picture that the DNA is just one single circle, and they sometimes have smaller circles of DNA as well. Eukaryotic cells, think eukaryotic are in you. These are the cells found in plants, animals, protists. Protists are things that live in pond water, like paramecium, and fungi, which are mushrooms. The eukaryotic cell is composed of four main parts. The cell membrane, which is the outer edge, and again that allows things in and out of the cell. The cytoplasm, which is the stuff found in between the different other parts of the cell. The nucleus, which would be here, which is the control center of the cell and houses the DNA. And all of the other organelles, which are the small structures that carry out specific functions. You can think of organelles like little organs. Now let's talk about some of the individual organelles. The nucleus is one of the most important. It acts as the control center of the cell. Notice even the nucleus has several parts. Nuclear pores, or holes in the outer membrane of the nucleus, which allow proteins and DNA to go in and out of the nucleus. The nucleolus, which is kind of the center of the center, and the nuclear envelope, which is a membrane just around the nucleus itself. It's usually found at the center of the cell because things need to go in and out, and so it needs to be relatively close to everything else. It has a nuclear membrane and nuclear pores. It contains the cell's DNA in one of two forms. Either chromatin, which is DNA bound to a protein, in a non-dividing cell. So this would be in a cell that's not actually in the process of dividing, or in chromosomes, which are the condensed DNA structures. They look like X's, and you find those in cells that are going through the process of division called mitosis or meiosis. It also contains an organelle called the nucleolus, which makes the cell's ribosomes. Ribosomes, again, you'll hear me say over and over, these are the protein-making things. The next organelle is the mitochondria. This is the cell's energy center. It turns the food you eat into a chemical energy called ATP. It turns it into an actual molecule. It's sometimes called the powerhouse of the cell. And you'll notice it, if you look in diagrams of the cell, it looks like a kidney bean, and if you look inside, it has all of these folds. The folds are where the chemical reaction takes place that forms the molecule of ATP. The Golgi apparatus. This is a folded structure as well. It processes, packages, and secretes proteins, and it's comparable to a factory or a post office. So it gets the raw materials inside these folds here, it puts them together, and then it forms a package around it. This actually breaks off, like you can see happen down here, and the proteins inside then move off to other parts of the cell where they are used for whatever their purpose is. These things that break off are called vesicles, and they're used to transport substances outside the cell. So things from the Golgi apparatus can actually go to other cells, neighboring cells. The lysosome is kind of the stomach of the cell. It takes down and breaks things down. It has digestive enzymes in it. Remember, an enzyme is a protein, and proteins are made in ribosomes. Sometimes they're called the suicide sacs. Another organelle is the endoplasmic reticulum. This is the intracellular highway. It moves things from various places in the cell to other places. There are two kinds. Rough endoplasmic reticulum contains ribosomes and is involved in protein synthesis. So if you look, here's the nucleus, or the edge of the nucleus. Here is the ER, and all those little dots on there are the ribosomes. The ribosomes, remember, are made in the nucleolus. And then you have smooth ER, where it's same sort of structure down here, but there are no ribosomes on the smooth ER. The cytoskeleton helps maintain support and shape. In the picture over here, the cytoskeleton is these tubules that you find here, these ones along here, and to some extent these things that kind of look like nerves. They support and shape the cell. They allow for movement. Microtubules are the hollow structures that help build cilia and flagella. Microfilaments are thread-like, and centrioles, which are only found in animal cells, and they're used during cell division. The next organelle is called the vacuole. This is found, it's found in both plants and animals, but the vacuole in plants is usually very large and in the center, and in a plant it's used for support and structure. But a vacuole is a storage area for water and other substances. If the vacuole in plant cells loses its water, then the plant wilts and gets all droopy. The cell is like a factory. Its main job is to produce proteins. Remember, proteins are produced in the ribosomes. These proteins then go to the body to serve different functions, different jobs. The DNA contains the instructions to build all the different proteins. The instructions are sent to the ribosomes. Remember, the ribosomes are made in the nucleolus, and they're found along the rough endoplasmic reticulum. The ribosomes build the protein and send it through that internal highway, that endoplasmic reticulum. And then those proteins are delivered to the Golgi apparatus, where they are packaged for export outside the cell. This is an example of a typical animal cell diagram. All of the parts that we just mentioned are located here. And remember that cells are three-dimensional things. It's not a flat disk. An animal cell is more like a beach ball that's not blown up all the way. So it has a variety of shapes. It's not just a sphere, but oftentimes they are spheres. This is an example of cheek cells seen through a microscope. We actually do this in 7th grade. The students take a swab of their cheek cell, rub it on a microscope slide, and this is the image that they see. So you can see that they're not perfectly round, and of course they've been broken a little bit because of the process of getting them out of your mouth. But this large dark area here is the nucleus. And then all of the other tiny little dots you see around there are the other organelles and structures within the cell. Plant cells have some additional structures, things that animal cells do not have. The first is a cell wall, which surrounds the membrane. So outside edge is the membrane, and then outside of that is the cell wall. Provides additional support so that plants can actually stand up as opposed to falling over. They don't have the skeleton that animals would. The next is a chloroplast. These contain the green pigment that make the plant green and allow the plant to do photosynthesis. And that central vacuole, which is the large water container in the center of the cell, which also aids in keeping the cells functioning properly, as well as keeping the plant standing upright and tall. So here you see, here's the central vacuole. It's kind of pushing out along the outside edges. These are the chloroplasts, which we'll learn more about when we study photosynthesis. They also have mitochondria. They have smooth ER, rough ER, ribosomes, Golgi apparatus, nucleus, nucleolus, DNA in there, lysosomes. All the other things that the animal has are also contained in the plant, as well as some other things. This is an actual microscopic view of Anacharis, or Elodea, which is a water plant. You can see here, these little green guys floating around are their chloroplasts. So remember that when you're talking about a plant cell, it's not just one chloroplast, it's many, many of them. And then you can see that the walls of this plant cell are slightly thicker. The membrane you can't really see, but it would be found right underneath the wall. Two of the organelles, mitochondria and chloroplasts, actually have their own DNA, which is separate from the DNA that's in the nucleus. And this is thought to have come about because if you study this diagram that you see here, basically it's telling you that scientists believe way back when there were only prokaryotes existing in time, that the prokaryotic cell kind of engulfed some of these other cells. So it's essentially like saying that the mitochondrion and the chloroplasts existed by themselves as their own cell, and then they got engulfed in a different prokaryotic cell and kind of adapted to make then the eukaryotic cell that contained these other things. Last but not least is the cell membrane, extremely important structure. It contains proteins, which kind of go the whole way through the cell membrane, and phospholipids. We learned about lipids as a macromolecule. Phospholipids, there's 2 layers of them. They've got the head of the phospholipid on one side and the tails on the inside. And this allows the cell membrane to be selectively permeable, which means it regulates what comes into the cell and what leaves. Again, composed of a double layer of phospholipids with proteins embedded throughout. And the proteins allow bigger things to go in and out of the cell. The phospholipids allow molecules that are slightly charged to basically just slip in between the layers and go in or out. If you have any questions on this assignment, please feel free to message me, and hopefully this will be an easy one for you.